So we left off last time talking about the prokaryotes, bacteria and archaea. We talked about what they are. We talked about the various reasons why we should care about them and the fact that they're super abundant and very influential because of their abundance. We talked about when they evolved in the sort of deep geological history of Earth. And last time we talked about why they're so successful, so why they're so abundant. And part of that is their metabolic diversity enables them to survive in a lot of different habitats. And another thing that we're going to talk about today for why they're so successful and why they've been able to evolve all these different types of metabolisms, as well as other adaptations that have helped them survive in a lot of environments and places where other organisms haven't, because they are the quote unquote masters of evolution. So let's talk about what I mean by the masters of evolution and why they've been able to evolve so many strange and different traits that other organisms and organismal groups have not been able to. So the first reason is because they've just been here the longest. So there's just been more time for selection and evolution to occur. So if you remember this figure that we looked at here, where we looked at the origin of Earth about 4.5 billion years ago, and about 3.75 billion years ago, we see prokaryotes, this yellow sort of bar here, jump onto the scene. And so in general, they've been around for almost a billion years longer, or I'm sorry, even more than that, than eukaryotic organisms. And so what you can see is there's just more time in general for evolution to occur. Many more generations of prokaryotes have occurred over this period of time. The second major reason is their fast rate of reproduction. So they're able to reproduce really quickly. That results in more generations per unit time and more opportunities for selection to occur, and also for novel mutations to arise. And so in essence, they have faster rates of evolution just because they're going through more generations in a shorter period of time. And I'll talk about this in a little bit more detail. And the third major thing is something called horizontal gene transfer. So horizontal gene transfer has also enabled prokaryotes to evolve such a diverse array of adaptations because they can exchange genes with others, which in the end ultimately increases the genetic variation that exists in populations of eukaryotes for selection to pick and choose from. So let's talk about this idea of a fast rate of reproduction and why this would enable prokaryotes to have such a diverse array of adaptations. So if we just think in general terms of prokaryotes' ability to reproduce like crazy, for example, E. coli can reproduce every 20 minutes. So compare that to a human that reproduces, every 20 years or so. E. coli can reproduce every 20 minutes. And if you do the math on that, every 48 hours, if that population of E. coli could grow unfettered, it would result in a population of 2 times the 144th power of E. coli, which is significantly greater than the number of humans that have ever lived on Earth in the entire history of humanity. And E. coli, in theory, could produce a population that big in under 48 hours. Now, obviously, E. coli doesn't produce populations that huge because there's limiting factors, there's selection, there's things that are stopping the population from growing completely out of control like that. Things eat and kill E. coli bacteria. And they have to compete for resources. But the point here is they can grow and reproduce like crazy, and this can have substantial effects on evolution. So if you recall, generation turnover is a key factor in the rate, quote unquote, of evolution. So the more generations you have, the more opportunities you have for evolution to occur. So if we think back to this Blueville example, and you remember that the definition of evolution is a change in allele frequencies in a population over time, to have a change in allele frequencies over time across generations, you have to go from one generation to the next. So for example, this first to second generation, there's no change in allele frequencies, and then in second to third generation, there is a change in the allele frequencies of the population after this selection event. And so if you can go through these different generations in a much quicker period of time, you can have more selection events, more opportunities for there be selection on individuals, and more opportunities for evolution to occur. And so the idea here is the more generations you have in a shorter period of time, the more opportunities you have for evolution. So given this, which of the following is likely to evolve the fastest? A, bacteria that has this generation time and this many generations per million years, or a dog or a cat, or C, humans? Go ahead and click on your answer. So if you said a bacteria, that is the correct answer. They have a generation time that's much, much shorter than either a dog or cat or humans. And if you think about the number of generations that occur over a million years, there's 87 billion to 365 million generations of bacteria, and potentially that many individual selection events happening over this million-year period. Whereas if you look at dogs and cats, there's about 500,000 opportunities for changes in allele frequencies or generations over time. And if you look at humans, there's only 45,000 generations over a million years. And so fewer generations, fewer opportunities for selection to alter populations of humans versus bacteria. And so given that they're able to turn over the generations really, really quickly, that gives rise to the question of how is it that they're able to do this? How is it that they're able to reproduce so quickly? And one of the first reasons is probably obvious, but in general, because they're so tiny, they really need fewer resources overall to reproduce in general. So it just requires fewer resources for them to make copies of themselves. Another way that they reproduce so quickly is their actual process of reproduction, which is a type of asexual reproduction through a process called binary fission. So I want to talk about how prokaryotes reproduce through this process of binary fission. So binary fission is one type of asexual reproduction. Remember, asexual reproduction is when you reproduce without using sex. So you don't have egg and sperm, you don't have males and females. So asexual reproduction is when offspring get all of their DNA from one parent. So one parent produces offspring, and they pass their DNA off to those offspring. And in this case, there's no gametes, for example, egg and sperm, although there are other types of gametes that exist out there, but no gametes in general are involved. And this is done by prokaryotes, as we talked about before. Archaea and bacteria both reproduce asexually, and some eukaryotes can actually reproduce asexually as well, although most eukaryotes reproduce sexually. And so prokaryotes reproduce through this process called binary fission. So this is a specific type of asexual reproduction. There are actually many different types of asexual reproduction. This is the specific type that prokaryotes do. And this figure here gives you a basic schematic diagram of how binary fission works. So let me walk you through the process of binary fission. Starting here on the left side, you have a parent cell. That parent cell's chromosomal DNA is replicated, so you make an exact copy of the DNA, and then the DNA copies migrate to opposite ends of the cell as it elongates, and then eventually the cell membrane and cell wall pinch in on that the two budding daughter cells until eventually there is this process of fission where the two new daughter cells separate. An important thing to pull away from this is that these two daughter cells, by definition, are going to be genetically identical to one another, and they're going to be genetically identical to the parent cell, assuming that no mutation occurs when the DNA is being copied, okay? And another important point to drive home here is this is an example of what's called vertical gene transfer. So vertical gene transfer, so remember vertical is from top to bottom, and so vertical goes from the parent to the offspring, from the top to the bottom, okay? So the parent is passing their DNA off to the offspring. This is vertical gene transfer. And the huge benefit of this binary fission is, as we talked about before, it enables prokaryotes to reproduce really, really quickly. And there's a lot of advantages to being able to reproduce very quickly and have a short generation time. In addition, when you're not having sex, you don't have to look or find a partner to reproduce with, and you don't have any of the sort of hangups that are involved with mating and breeding with another individual. You can reproduce, or bacteria can reproduce on their own. So it simplifies reproduction. But it creates this issue of how is genetic variation introduced. So remember that genetic variation is extremely important for natural selection, because natural selection needs genetic variation, or it needs variation in traits in a population, and those variation in traits ultimately derives from genetic variation in the population. And if you have daughter offspring, like these over here, that are genetically identical to the parent, how do you ever get any new genetic variation in the population in the first place? So one place and the ultimate source of all variation in any population or any individuals, whether you're eukaryotes or prokaryotes, is through mutation. And so any new trait that appears in a population um, ultimately has to come from some mutation in the genetic makeup of individuals in that population. So you can have new traits come in from a different population, but those new traits still had to ultimately evolve from mutation. So when I say the ultimate source of all variation is mutation, that means like if you find a new trait in a population and you trace it all the way back to where it first came from, ultimately it came from mutation. The other place that prokaryotes can get genetic variation or generate genetic variation is through horizontal gene transfer. And this is of traits or alleles that are already extant in a population or a different population somewhere. So these are not brand new traits that are introduced by mutation. These are traits that have already been introduced by mutation and then get passed around to different individuals or different populations through this process of horizontal gene transfer. So this is the third sort of major reason that prokaryotes have evolved such a diverse array of adaptations and they're the masters of evolution is because of this process called horizontal gene transfer, where individuals can exchange genes with others. So let's talk about horizontal gene transfer in a little bit more detail. So before we do that, we need some background or some reminders on where bacterial DNA exists and the different forms it exists in bacterial cells. So most genes in a bacterial cell are held on sort of larger circular chromosomes. So if you can remember this figure out we looked at before, and we're looking at these sort of like dark blue spaghetti stuff, this is the chromosomal DNA that's floating around in a bacterial cell. And this is where the vast majority of the genes that bacteria have exist. But bacteria also have these things called plasmids, and some genes exist on plasmids. And so it's this light blue sort of hula hoop thing that you're looking at right here represents plasmid DNA. And plasmids are typically smaller circular pieces of DNA, so typically less than 1/20th the size of the typical chromosomal DNA. And they can replicate independently of chromosomal DNA. And these plasmids are pretty common in bacteria. So for example, in E. coli, so like all of the different populations of E. coli all over the world, as many as 3,000 different plasmids have been identified in different individuals. And some individuals can have multiple plasmids. And plasmids, as I said before, typically carry genes on them, but the genes that they normally have are only required under usual circumstances. So things like antibiotic resistance genes are typically found on plasmids. So for example, here we're looking at chromosomal DNA, here we're looking at the plasmid DNA, and if we zoom in on one of these plasmids that's in this bacterial cell, we can see that this plasmid has a gene on it right here for antibiotic resistance and maybe one other gene. And so they have very few genes, and most of those genes are not genes that are required for everyday cellular processes that the cell needs to survive. They're only needed for unusual circumstances, okay? So typically, the chromosomal DNA has what's called the housekeeping genes that are needed for everyday survival and reproduction. So things that the cells need to do every single day, like producing proteins, like getting rid of waste, like producing RNA, and all the sort of stuff that a cell needs to do on a regular basis. So you can think of chromosomal DNA having those housekeeping genes that are being used every day, in and out, and plasmids having genes that are only used in rare circumstances. For example, when an antibiotic gets introduced to this bacterium and it's like, oh man, things have changed really significantly, better break out the plasmid genes. Okay, so given that background, let's talk about horizontal gene transfer, or transferring DNA from one parent bacterium to another. So the idea here is with horizontal gene transfer, you can have a donor cell and a recipient cell, and the donor cell can basically transfer some plasmid DNA to that recipient cell. Sometimes you can also get chromosomal DNA going into that recipient cell. But the beauty here is that these individuals don't need to be related to one another, so this is not the same as sex. They don't even need to be the same species. It's sort of the equivalent of if, a human could walk up to another human, shake hands with them, and say, hey, I'd like to have your curly hair. and all of a sudden you've got that curly hair trait. So they can just sort of pass traits off from one individual to another horizontally. So not from parent to offspring, but from individual to individual. And there's three major mechanisms of horizontal gene transfer. And it's important to note that none of these occur during binary fission. So this is not happening while binary fission goes on and bacterial cells are reproducing. These things happen independently of binary fission and can happen at any time sort of in the life cycle of a bacterium. So one is transformation, where bacteria take up free DNA released by other bacteria. So essentially one bacteria dumps some DNA out into the environment and another bacterium picks it up. Another is transduction, where DNA is transferred from one bacterium to another by a virus. So a virus basically infects one bacterium, sucks up some of its DNA, and then infects another bacterium and inserts some DNA into that second bacterium that it infected. And you can also have conjugation, where essentially there's bacterium to bacterium contact and DNA is transferred between those two bacteria. So let's just watch some quick videos about how this stuff works.
Bacteria and archaea generate genetic variation, in part by transferring DNA from one organism to another, sometimes between individuals that are only distantly related. In a process called conjugation, one bacterium forms a long tube called a pilus that attaches to another cell, enabling DNA, usually a plasmid, to pass into the second cell. In transformation, DNA breaks apart after cell death and is released into the environment. A second cell takes up strands of DNA and incorporates them into its genome. In transduction, segments of bacterial DNA are incorporated by viruses and transferred to new cells by viral infection. Bacteria and archaea.
And so just to sort of put all this into the big picture, this idea of how reproduction relates to the transfer of genes from one individual to another or from parent to offspring and ultimately results in genetic variation in the population. Remember, the big idea here that we're always trying to get at is how do we create genetic variation in a population? And that genetic variation is super, super important for natural selection and evolution to occur. Because if you don't have any genetic variation in a population, they're not going to have different individuals to natural selection to choose from. So it's super, super important that organisms have ways to generate variation in their populations. And so if we look at vertical gene transfer, which is transfer of genes from parent to offspring, and horizontal gene transfer, which is essentially transfer from individual to individual, which could be considered parent to parent or adult to adult, or even offspring to offspring. The idea here is if we look at eukaryotes cannot do horizontal gene transfer. They can only transfer genes vertically. And so the way that eukaryotes generate genetic variation through this reproductive process, the way that they produce genetic variation in their offspring is through this process of sex. And so if you can remember back to meiosis in the way that sex works, you have mixing of genes from different parents mixing together to produce new combinations of genes in the offspring. And those new combinations of genes and alleles in the offspring results in genetic variation in the population. And you can have continuing mixing through the population over many, many generations. And so this creates the genetic variation that natural selection needs to select from. Whereas if you look at prokaryotes and the way that they reproduce for binary fission. And by definition, binary fission results in genetically identical offspring. So that's true unless there's a mutation, but those mutations are very, very rare. And typically, even if you do have a mutation, the first thing that happens is that offspring dies immediately. And so it doesn't result in new variation in the population. And so the question is, how do you generate the genetic variation that's needed in a population if these prokaryotic organisms are producing only by binary fission and always producing genetically identical offspring? Well, the answer to that is through horizontal gene transfer. So prokaryotes, although they produce by binary fission, they have a different way of introducing genetic variation into their population, and that's through horizontal gene transfer. And this can result in lots of new gene combinations in the bacterial populations and tons of genetic variation for natural selection to work on. Okay, so just to reiterate, how have prokaryotes evolved such a diverse array of adaptations that we've talked about in previous lectures? One, they've been here the longest. Two, they have more generations per unit time, so they basically, evolution works a little bit faster in them. And three, they can have a ton of genetic variation introduced into the population through horizontal gene transfer, or the exchange of genes between individuals. And so ultimately, this faster reproduction, horizontal gene transfer, can allow advantageous traits to arise and spread more rapidly through these populations, allow bacterial populations to adapt more quickly, and this is why prokaryotes are the masters of evolution. So, a quick question. Which of the following statements is true? Okay, so if you said prokaryotes can transmit genes via vertical and horizontal gene transfer, that is correct. So remember, prokaryotes can send genes from parents to offspring via vertical transmission. They can also send genes from parent to parent through horizontal gene transfer. B is false because mutation is the ultimate source of genetic variation in both eukaryotes and prokaryotes. So if you take any new allele that you haven't seen before in a population and ultimately trace it back to where that new allele first arose, ultimately you're going to go back and find that that new allele evolved from a mutation. Okay, so all new traits, ultimately, if you follow them all the way back to where they first came from, whether you're in a eukaryote or a prokaryote, come from mutation. It's just the way that those traits can be spread and shared over generations differ from eukaryotes and prokaryotes, where eukaryotes can spread those traits around through sex and mixing, and prokaryotes send those traits vertically through binary fission and can also share those traits through horizontal gene transfer. Typically, chromosomal DNA carries the housekeeping genes, not plasmids. Plasmids really carry genes for rare circumstances like antibiotic resistance. Transformation, conjugation, transduction, and mutation all play an important role in generating genetic variation in prokaryotes, but meiosis and sex are unique to eukaryotic organisms. Next question. Okay, so if you said, why is genetic variation important for prokaryotes? That is because it ultimately leads to the variation in traits that's necessary for evolution to occur. So you need to create genetic variation for natural selection to have different variants to choose from and ultimately for evolution to occur. So the last thing I want to talk about is sort of what are the ultimate outcomes of horizontal gene transfer and this ability of DNA to mix their DNA very, very frequently. And there's good outcomes from horizontal gene transfer. Specifically, you can have the evolution of a lot of beneficial adaptations that we talked about before. So these different metabolisms that are vital in nutrient cycling, like we talked about in previous lectures, like the nitrogen cycle. In addition, the ability for bacteria to do bioremediation. So once again, having these diverse metabolic processes allow bacteria to eat up oil, or also to produce biofuels photosynthetically. And so these are all crazy sort of metabolic abilities that have evolved through the ability of these bacteria to evolve interesting and novel traits. In addition, the beneficial bacteria that we have all over our microbiome, so things that can help produce antibiotics, immunity, and nutrients. Once again, these are all traits that have evolved ultimately through mutations and the spread of those mutations through horizontal gene transfer. But there's also some bad sides of the ability of bacteria to do horizontal gene transfer. And one of the most notorious examples of that are the transfer of antibiotic resistance genes in plasmids. So plasmid transfer can lead to multi-drug resistant virulent bacteria. So for example, you can have plasmids that have genes for antibiotic resistance in one individual donor cell. Then that plasmid can be transferred to another bacterium of the same or different species. And what that can lead to is multi-drug resistance in some bacteria. So for example, We see an extensively drug-resistant tuberculosis. It's XDR-TB. Basically, this is a strain of bacteria that's essentially resistant to all of the most effective tuberculosis drugs. And you have one bacterium that's harboring multiple plasmids that has antibiotic resistance genes. Um, and those plasmids and those bacteria can spread to infect different individuals. And on top of that, they can even be selected for if those individuals are given antibiotics and these bacteria are present in smaller amounts. They can be selected for because the other strains of this bacteria die. So the idea here is that this horizontal gene transfer can lead to bacteria that have multiple plasmids that have resistance to different types of antibiotics, resulting in extensively drug resistant strains of bacteria that can be very difficult to deal with because we have to develop new antibiotics to be able to wipe those bacteria out.